Yes, good day, viewers. This is 300 Plus Academy, where all we do is exams tutoring, ensuring that you ace your next exam. I am Adeyemi Abiodun, the lead tutor at 300 Plus Academy. Come along with me into the world of biology. The last time out, we had lectures in biology. We were talking about introductory biology, where we looked at biology as a decathlon of science, telling you that biology involves various fields. It's an enormous subject, and also looking at defining biology and, of course, the characteristics of life. But today, what do we have to serve you? It is the very important business of cells, talking about cytology. So, cytology, briefly, is the study of cells. So we'll be looking at cell biology, and I have some outline here, which is in tandem with what our UTME syllabus entails. First, we're going to be doing a brief introduction, talking about cell biology or cytology. We'll be looking at cell theory and history. I will be discussing with you why cells are small. I will be, highlighting briefly on the microscope, talking about the light microscope and the electron microscope, and looking at the resolution problem as to why is it that I cannot see cells? Why is it that you cannot see cells with the unaided eye? We'll be looking at that, and as well talking about complexity, in living things, and as well as what we call surface area to volume ratio. That is what I mean here by surface area to volume ratio, because talking about complexity and surface area to volume ratio, these are precursors for us to be able to understand why we need a transport system, why we talk about respiration, and talking about functional biology as it is. We'll be looking at prokaryotes, otherwise known as prokaryotic cells, and eukaryotes or eukaryotic cells. Then we'll take a tour of eukaryotic cells, talking about try to understand that machinery. Yes, I call it machinery called cells. We'll be looking at the variety of cells we have. We'll be looking at the various forms in which cell exists. We'll be looking at organization of life, because life exists in an organized form, from a simple atom to a molecule, to complex molecules, to organelles, to having a cell, and then to having tissues, to having organ, and to having systems, and then to having the organism. So we'll be looking at how life exists. Everything is not chaotic. Everything is not in disarray. It is existing in an organized form. We'll be looking at that. And also, very importantly, we will be looking at how substances move into the cells and how substances move out of the cells, because the right materials must get into the cells and waste materials must move out of the cells if the cells are to be kept alive. When my cells function, I will function. When my cells are alive, I will be alive. And like I do say, the only thing I cannot do without are the cells of my body. I actually can do without anybody. So there is no such thing like without you, there will be no me. Oh, I can't do without you. No. You can do actually without your boyfriend. You can do without your girlfriend. The only person you can do without are the cells of your body, because they are the basic structure and they are the functional unit of life. When they function, you function. When they live, you live. When they die, let's stay alive. Don't die. Stay with me. Stay with me. How do you stay with me? Hit the subscribe button. Turn on the notification bell. Tell your peers, your boyfriends, your girlfriends, your enemies, your friends. Tell them this is the place to be to ace your UTME exams. And I tell you, you won't regret it. So let's move on. So moving on, when we talk biology, you recall that we said biology is the study of life. So the next sensible question to ask is what is life? And we said then that, it is not actually possible to define life. That is not an easy task. The best we can do is to list features or characteristics exhibited by living things. But this time, we are looking at characteristics that are central to life. We are looking at characteristics that are exclusive to living things. When you talk about characteristics that are more central to life, take reproduction for instance. That is not central to life, as not all living things reproduce. So we cannot use the criteria of reproduction to define life. But when you talk about excretion, all living things excrete. That is more central to life. When you talk about cells, all living things possess cells. So that is more central in defining life. When you talk about irritability or sensitivity or responsiveness, that is more central to life because all living things respond to stimulus. When you talk about things like movement, that is not central to life because non-living things also move. When you talk about growth, increase in size, that is not central to life because non-living things also grow. When you're talking about features that are central to life, like we said while we were talking about the features of living things, you're looking at things like excretion, you're looking at things like metabolism in living things, you're looking at things like adaptation, you're looking at things like inheritance. What are the traits I have inherited from my parents that gives me the features that I have? You're talking about those special molecules like DNA, deoxyribonucleic acid, RNA, that codes for all of the features that I exhibit. Inheritance is more central to life because I have inherited certain traits from parents. You have inherited certain traits. So when you talk about things that define life, you're looking at features or characteristics exhibited by living things but not exhibited by living things. But one of such features which is very, very key and exclusive is the possession of cells. All living things possess cells. That I need you to understand first. We said that, and talking about these exclusive features of living things, the link to that lecture video is going to be in the description to this lecture. In case you need to go over it or you miss out on that, this is the right time for you to quickly go through that and refresh your memory on that. So moving on, I do ask, anywhere I've lectured on cytology, I do ask, in what part of you as a living thing or as an organism, in what part of you is the life? I am alive. I believe I have life inside of me. So I do ask, in what part of me, in what part of you does that life exist? I asked a student not so long ago. The student mentioned the heart. Another student said the brain. Oh well, sorry to burst your bubble. That is not what I'm expecting. And I like to use this analogy. If I say a car, for example, what is the most important part of a car? What makes a car a very good car? The student said the engine. And that is true. If the engine functions, the car will function. There is no point having a beautiful car with a very bad engine. There is no point having a beautiful car that cannot travel distance because the engine is faulty. It is the same way with you. You ask yourself, what is my own engine? What are the engines that power living things? You ask yourself, what are the engines that powers you? I ask myself, what are the engines that powers me? The engines that powers me are the cells of my body. Just as the engine is to the car, so are the cells to living things or organisms. So when you talk about where the life I possess so when you talk about where the life that I possess is inside of me, the life I possess is actually inside the cells of my body, and that's just that about that. So because I want to study life, because I want to maintain life, it is only imperative that I understand what my cell is like, that I understand how my cell function, because for me to maintain life, it now means I need to maintain the cell. That said, so let's quickly define cell. When we say cells, cells, yes, we do say is the basic structural and functional unit of life, which simply implies that when the cell functions, living things will function. And also, it means that when the structural integrity of the cell is intact, then living things structurally will also be intact and function well. Also, we can define cells uh, as tiny microscopical units, which means we cannot see them with the unaided eye. But you see the cells, they collectively carry out the processes that makes the organism a living entity. One of the things we must know about the cell is that even when we cannot see them, with the unaided eye, you need to understand that the cells are alive, with activities buzzing inside them. It will surprise you the amount of activities that goes on inside the cells, even without we noticing. But all these processes culminate into making the organism a living entity. Also, it is important to know that most microorganisms are usually made up of single cell, a single cell, that's unicellular, talking about amoeba, paramecium, cordatum, euglena viridis, chlamydomonas. These are examples of a single cell. Also, let me make you understand that the yolk of an egg, although large, and in this case we can see it, but the yolk of an egg is actually one enormous cell. It's a single cell. Talking about the yolk of an egg, it's a single enormous cell. Talking about most plants and animals, you need to understand that we are made up of many cells. In other words, multicellular. Talk, for instance, humans, we are made up of about 60 trillion interacting cells. So that is that on the definition of cells. So the next thing we want to talk about is fact about cells. That's talking about the cell theory and its history. Moving into the cell theory, it simply means we want to look at facts about cells. Of course, the first fact is always that cells were first observed by Robert Hooke in the year 1665. He observed thin slice of cork. He observed thin slice of cork under a light microscope, and what he saw then were numerous bus-like structures. He saw bus-like structures, as you can see on your screen, and he called those bus-like structures little rooms. These are small compartments, which are then later named cell. That was all he saw. He didn't move further or examine things further by checking the content of those little rooms, of those tiny compartments he had seen. All he did was to just name those compartment cell, probably based on the tools available at that time, compound microscope. The tools available were not all that sophisticated, and that was why we said there he saw many small compartment little rooms, which he then named cell. Here, Hooke did not pursue his discovery further by investigating the structure and function of living cells. Of course, probably limited by the crude set of tools available at 1665. Also important to know that the cells that Robert Hooke observed were not actually living. Why are they not living? Very simple. Recall that what you observed were actually thin slices of cork. When you talk about cork, cork is the outside part of the stem of a plant, as you can see in the diagram on your screen. Why are those cells not living? Look at it this way. If this is a young plant, as the plant grows, it simply means that new cells will be formed in the plant via cell division or mitosis. So let's look at a case of, the cells of this young plant dividing rapidly or multiplying rapidly, such that the plant had grown in size this way. You need to understand that the new cells will be around here, the new cells will be around here, whereas the old cells will be pushed out. The old cells will be pushed out, so that the cork cell we are talking about are actually those group of cells on the outside part of the stem, on the outside part of the stem. So what happens is that as new cells are being formed in the plant, the old cells are being pushed to the outside, and as they are being pushed to the outside, they become filled with a waxy substance, called suberin. They are filled with a waxy substance called suberin. Because this substance is waxy, it simply means it then renders the plant cells impermeable to water, and consequently the plant cells die. And as they are even being pushed further out and new cells are being formed at the center, they become converted into wood. They become converted into wood via a process called lignification. When we say lignification, it simply means, in this case, as the dead cells having been filled with a waxy substance, suberin, which renders the cell impermeable to water, it simply means in this case that they become hardened, they become converted into wood, because a substance called lignin, in this case, is added in the plant, hardening the cells and converting them into wood. So when you look at the fact that Robert Hooke only took, thin slice of cork, that's the outside part of the plant stem, what he was basically observing were just dead cells that are no longer active. Hooke did not even pursue further his discovery to realize the significance or the importance of the cell. The only thing said there was that I had observed cork, a thin slice of cork, and I saw numerous bus-like structure, as you can see displaying on your screen. That is as at 1665. The first living cells were observed by Antone Van Leeuwenhoek, a Dutch microbiologist in 1674. You look at that, 1665, 1670. But that's just some years later. In this case, he used, of course, a microbiologist, he used a much improved microscope. And what he observed at this time was a free-living algae spirogyra. He observed that in a pond water, and the organism he saw this time, he called that tiny organism animalcules, meaning little animals. So this is what he had observed using a much improved microscope, and he called that animalcules, just like particles, in this case tiny or little animals, animalcules. And that was it as of then. Not Robert Hooke, not Antone Van Leeuwenhoek still realized the significance of the cell. It took 200 years later, of course, much improved tools, much improved instruments, for Felix Dujardin, who then propounded then that most microorganisms are made up of single cell. Felix Dujardin propounded then that most microorganisms, when you say 200 years later, we are talking 1835. At that time, Felix Dujardin wrote that most microorganisms are made up of a single cell, and he checked that the internal substance, the internal content of most of these living cells are similar. Other scientists, of course, Matthias Claydon, Theodor Schwann, Rudolf von Moll, just to mention a few, all one way or the other contributed to the development of the cell theory. So you want to look at it, the cells that Robert Hooke observed were not living. Then Antone Van Leeuwenhoek, much improved microscope, couple of years later, observed a free living spirogyra in pond water and called the tiny organism he had observed animalcules. 200 years later, 1835, Felix Dujardin said, look, most microorganisms are made up of cells, and also found out that the internal content of most of these living cells are alike in many aspects. Other scientists, Matthias Claydon, Theodor Schwann, try to put this, into a fact, try to arrange this into a fact, which we now know as the cell theory. Rudolf told us then about dividing cells, rapidly dividing cells, talking about the fact that new cells arise from pre-existing cells. That's just to, talk about some of the works of those scientists that had contributed to the cell theory. So to put things in proper perspective, when we say the cell theory, the first thing is that all living things are made up of cell or cells. In other words, cells are of universal occurrence. Secondly, the cells are the basic structural and functional units of life. And thirdly, new cells arise from pre-existing cells. At that time, Rudolf wrote that the body is like a state in which every cell is a citizen. And of course, if every cell is a citizen, new cells can only be formed from pre-existing cells. In this case, new cells are formed via cell division, or what we call mitosis. So that is just that about the cell theory. So looking at these and many other things, to put things into writing, to put things into proper perspective, the cell theory states that one, cells are of universal occurrence. This means that all living things are made up of cell or cells. When we say cell, we're talking unicellular. When we say cells, multicellular. But the important thing is that any living thing that you see is made up of cell or cells. That cells are of universal occurrence. Secondly, cells are the basic structural and functional units of life. They are so central to life. When they are alive, the organisms will be alive. A lot of activities is actually going on inside them that keeps us alive. So they are the basic structure and the functional unit of life. And thirdly, as stated by Rudolf Virchow, he wrote then that the body is like a state in which every cell is like a citizen. And if every cell is like a citizen, a new citizen will only come from a pre-existing citizen in that state. In other words, new cells comes from already existing cells. That's pre-existing cells in that living thing via a process called cell division or mitosis, which much later we're going to look at mitosis under growth, talking about process of cell division, interphase, prophase, metaphase, anaphase, and telophase. I did back then use this acronym to store that, get that locked upstairs using the acronym IPMAT. That's one of the things we do in biology. We just use an acronym to represent an array of processes. But the important thing here is that new cells arise from pre-existing cells via a process called cell division or mitosis. So this is the fact about cell. This is what we call the cell theory. Why is it that cells are small? I'm coming back to the resolution problem. Why is it that we cannot see the cells with the unaided eye? talking about why we cannot see the cells with the unaided eye. And of course, I'll try to link up complexity with this. Now, let us imagine that, okay, let me talk about two scenarios here, A and B. Okay, let this be some group of cells here. Let this be some group of cells, and let this be some group of cells here. So when you look at cells in group of cells in the case of A and the cells that make up B, you're going to see, so let's assume that of course we have a material outside here that needs to move into the cell, and we have a material here that needs to move into the cell. We have a material here that needs to move into the cell. Where do we think the materials will get into the cells very quickly? It is going to be in the case of A. Look at it. The distance to move here is short. The distance to move here is more. So if the right materials in this case are to move into the cells, the right materials will move in with minimum difficulty. Unwanted materials or waste materials will move out of the cells with minimum difficulty because they now need to travel very short distance. But look at in the case of here, that's not going to be with ease. So exchange of materials, movement of materials in and out of the cells is with minimum difficulty in the case of A, but in the case of B, it is not with minimum difficulty. In that case, we call that biological communication. Cells are small for communication reasons. Communication reasons in this case talks about how the cells, interact with its environs now, talking about transfer of materials in and out of the cells. The smaller the cell size, the easier it becomes for the cells to release the right materials to the outside, and of course to communicate with other cells. Communication in this cells talks about interchange of materials. When a cell is small, such interchange is going to occur with minimum difficulty. But when a cell is large, look at it. Before these materials gets into the cell, and before a material moves from here, and before a material moves from here to here, you can see that here it's going to be with ease, it's going to be faster, and the distance to travel is less. Actually, I put it to you. Imagine having 100 small cell size and about 3 large cells. Where do you think we are going to have more effectiveness? The effectiveness is going to be in the case of having 100 small cells, because of the ease with which materials will move in and out. Unlike when I have 3, the ease with which materials move in and out is not going to be easy because you're talking of traveling long distance here. In the case of this, we call this having a large surface area to volume ratio, and here we are talking of having a small surface area to volume ratio. That is, when you look at the surface available for materials to come in just for these cells now, you're looking at a very large surface as compared to the size of the cells. And when you have a large surface area to volume ratio, diffusion is enough to take care of the exchange of materials. But when a cell is large, you're talking of a small surface area for the right materials to move in as compared to the volume. So in the case of this, we say cells are small for communication reason. In this case, biological communication, talking about transfer of materials in and out of the cells. Also, very important to note is regeneration of cells. Here it is easier for cell divisions to occur, or mitosis, and for new cells to be formed. Unlike in the case of B, regeneration is not going to be very easy here. As some of us know, if we don't know, we should know now that with complexity, with increase in size, a regeneration of lost parts in organisms becomes difficult. But with a very large surface area to volume ratio with less complexity, when cells are small, regeneration is very easy, and that is the tell you that a cell being small is actually an advantage over when a cell is large. No, having a small cell size is an advantage over having a group of cells with large cells, because of simply transfer of materials in and out of the cell. In other words, communication reason and regeneration of cells. It is easy to replace cells when the size are small as compared to when the size are big. So I said here, small cell size confers large surface area. That's large surface area for materials to move in and out of the cell as compared to the volume. Unlike when a cell has a big size, then the surface area to volume ratio is not going to be is going to be small. In that case, thus materials move in and out of the cells with minimum difficulty. Just explaining here that the fact that we cannot see cells with the unaided eye, in other words with the ordinary eye, tells us that the cell is small. So I now put it to you that the cell is small. Is it an advantage or a disadvantage? That the cell is small is an advantage because substances can now move in and out with ease, and regeneration of cells is easy as compared to when a cell has a large size. So that ends that. So since we cannot see the cell with the unaided eye, why is it that we cannot see the cell with the unaided eye? The answer is because of what we call resolution problem. Resolution problem. Resolution problem of the eye. Resolution problem of the eye. When you talk about resolution problem, if you look at a cell now, if you look at this cell, if I am looking at this with my eye, with the unaided eye, I'm going to see this as a single fused image. I'm going to see this as a single fused image. In other words, what that means is that what my eye is seeing is that this is fused. I am not able to see any distance here, like okay, to this side, to this side. It is my eye is not able to see this as okay, this point to the right, to the left is far apart. But in the case of a microscope can resolve this as a higher resolution, such that I am no longer going to be seeing this as a single fused image, but rather I'll be able to see this side and then this side. And if it were to be even more a powerful microscope that has very high resolution, then I'll be able to see the details. But the reason why I cannot see cell is because my eye has limited resolution. But for something like a compound microscope now, a compound microscope, for a compound microscope, it can magnify as much as 1,500 times of course. But when you talk about an electron microscope, you're talking of a resolution of 500,000 times. So when we say a resolution here, it now means that for here, my eye is seeing this as a single fused image. This could be, let's say, with a compound microscope, okay? With a compound microscope, it could be this, such that I'm now seeing this as this side and then this side. So that is resolution. But when you talk about an electron microscope, you're looking at a cell, you know, enlarged as far as this size, like 500,000 times, which means more resolution. So in a nutshell, the reason why I am not able to see a cell with my unaided eye is because of the limited resolution of the human eye. My eye has what I call limited resolution. Talking about resolution, resolution is the minimum distance two points can be apart. The minimum distance 2 points, the minimum distance 2 points can be apart and still be distinguished at two separated points. So what is the minimum distance here? That these 2 points here and here is apart and can still be distinguished as two separated points. But the important thing is humans cannot see cells aside, of course, the egg cell. We cannot see the cell with the unaided eye because our eye has what we call limited resolution. For a compound microscope or a light microscope, it could magnify or resolve to as far as 1,500 times. For an electron microscope, it could do that by at least by times 500,000 times. But there is a snag about the electron microscope in that the whatever it is we want to examine with the electron microscope, what we want to examine must be mounted on what we call a vacuum. That's that about electron microscope, but there is a snag, there is a problem about the electron microscope, as whatever it is that we want to observe or view with the electron microscope must be mounted on a vacuum. As powerful as the electron microscope is, there is a snag, there is a problem about the electron microscope, one of which is that whatever it is that we want to examine, whatever material we want to examine, has to first of all be mounted on a vacuum. A vacuum in this case is an empty space devoid of any material, and thus whatever it is we want to examine is actually dead before the examination. So that is that about that, talking about having a small cell size, of course, which we've said is an advantage. Talking about the resolution problem, looking at the magnification for compound microscope, electron microscope, talking about resolution itself, and looking at the problem with the electron microscope. We need to now talk about prokaryotes and eukaryotes, talking about prokaryotic cells and eukaryotic cells. When you talk prokaryotic cells, you're talking about the simple non-complex cells of bacteria. And when you're talking about eukaryotic cells, you're talking about the complex, well-organized cells of plants and animals. Like I said here, eukaryotes have distinct interior compartments. Prokaryotes have no distinct interior compartments. This here is the basic difference between the two. As you can see on your screen, you can see a prokaryote. No organelles, even no nuclear membrane, no central nucleus. What you just have is about the genetic material, just coiled around the center. That's otherwise called nucleoid in some cases. But when you look at eukaryotes have nuclei and they have nuclei membrane. That's they have the nucleus and they have a nuclear membrane that separates the nucleus from the cytoplasm. In fact, we can use this analogy. When you talk about prokaryote is like living in a one-room cabin where eating, sleeping, watching TV all takes place in the same one room. The implication of this is that in a one room, because there are no compartments, one activity will get in the way of the other activity. Imagine someone wanting to sleep in a one room and I want to listen to a very loud music. There will be distraction. And then let's say we have another person that wants to read. So the implication of not having a clear, distinct interior compartment or having an organized interior for a cell is that multiple biochemical process cannot take place. Multiple biochemical process cannot take place. Whereas if you look at eukaryotic cells, the mitochondrion bounded by its membrane, the nucleus bounded by its membrane, the lysosome bounded by its membrane, and the plasmic reticulum bounded by its membrane. The implication of this is when you say eukaryotic cell, it's like, say, living in a 5-bedroom, detached house. The implication is that so many activities can go on concurrently without one activity disturbing the other activities. And the implication here is that in here, multiple biochemical processes, multiple biochemical processes can take place. In fact, the hallmark of eukaryotic cell, what makes eukaryotic cell stands out and what confers on it its organization, its interior organization and complexity is compartmentalization. Compartmentalization is compartmentalization. You see, the compartment is what gives it its organization. Just for instance, now where I am having this class now, there are walls that separate where I am holding this class for what is happening on the outside. If there are no walls, if there are no compartments, the implication is that noise disturbances, from nearby is definitely going to get to me, which now means that this activity cannot take place because of interference. But because there are compartments, there are walls that separate this room from what is going on out there, that means this process can take place, whereas the process on out there can as well take place. I am not interfering or getting in the way of what is happening outside, and what is happening outside is not getting in the way of this class. So the implication is that for eukaryotic cell, as you can see how organized the nucleus is on your screen, the implication or the hallmark or what stands eukaryotic cells out is actually those compartments, or simply say compartmentalization. You look at your prokaryotes, the bacterial cells, everything is just, so simple. The cells are small, everything's scattered, ribosomes all over, yes. Then you have a central, nuclear material, not organized, not separated from the rest of the cell. The general cell organization, it's sort of simple. So let's look at more difference between prokaryotes and eukaryotes. For prokaryotes, no nuclear membrane, no nucleus. You just have a central nuclear material, otherwise known as nucleoid, at the center, not separated from the cytoplasm per se. But when you talk about eukaryotes, we talk of a nucleus and a nuclear membrane that separates it from the rest of the cell. When you talk about prokaryotes, they are small, they are simple, and most are unicellular. When you talk about eukaryotes, they are large and complex. The size for a prokaryote is from 0.1 micrometer to 5.0 micrometer, but for eukaryotes, much bigger, 10 to 100 micrometer in size. Talking about prokaryote, they lack membrane-bound organelles. You see the various little, when you say organs, organs means little organelles. When you say organelles, organelles means little organs in the cell, in the cytoplasm of the cell. When you talk about prokaryotes, they lack membrane-bound organelles, but eukaryotes here have membrane-bound organelles. Everything is organized on the inside. When you talk about reproduction, prokaryotes reproduce sexually and asexually, whereas most eukaryotes reproduce sexually. Cell division in prokaryotes is by binary fission. Binary fission, you know, where the cells just split into two, whereas most cell division here is by mitosis, is by mitotic cell division, talking about your interphase, prophase, metaphase, anaphase, and telophase. So that's just a distinction between prokaryote and eukaryote. Next thing now is that we are going to take a tour of eukaryotic cell. Don't forget that we've said biology is the study of the life. Don't forget that we've said biology is the study of life, and the life that we want to study actually exists in the cells. So it is now time for us to examine closely the cells of animals and the cells of plants, so that we look at the various parts of that machinery and how they function.